Okay, I'm posting the link to today's lecture in the chat. I moved things around a little bit. I decided that the logic of talking about genetic variation today didn't quite make sense yet without talking about DNA sequence alignment. So that's going to be the focus of today's lecture. We're going to talk about the high-level concepts and some of the algorithms used for mapping DNA sequences to a reference genome or to a transcriptome, and then aligning those sequences. And I think often the terms mapping and alignment are used interchangeably. They're actually different. So we're going to talk about the distinction. We're going to talk about some of the problems with DNA alignment and mapping, what aspects are, when it's easy, when it's difficult, and what some of the consequences are of problems or errors in DNA mapping and alignment. So today, this is going to be just a conceptual lecture. We're not going to be doing anything on the command line or R today, so you can sit back and listen again. And please interrupt me with questions if you have them along the way. So we haven't talked about a FASTQ file yet, but I'm going to tell you what it is. The next lecture we'll talk about FASTQ files. But FASTQ is the text file that you get out of a sequencer. So you put DNA into the sequencer. We'll talk about how sequencing technologies work next week. But you know, from a high level, you put DNA in and you get a file out. And this file format that is the starting point for basically any genomics experiment is called FASTQ. And essentially it's just a big, and you'll see, hideous file format, which tells you the name of the DNA sequence. So every one of the, say, billion fragments that you get out of a DNA sequencer will each have their own name. They'll have a DNA sequence associated with them. So that's the main substrate that we're trying to use. And then they'll have some quality scores associated with them. And that's really a reflection. Those quality scores indicate how confident the sequencing technology was that this nucleotide truly was a G or that one was truly a C. Unfortunately, these sequencing technologies make mistakes. Thankfully, they don't make mistakes all that often. So our life has gotten a lot easier. When I first started working with Illumina sequences, it was actually still called Celexa then. This was 2005. The error rate was quite high. It was something like 1 out of every 20 nucleotides that it predicted was wrong. Which, if you're looking for genetic variation, as we're going to talk about today, if the nucleotide sequence is wrong that frequently, you can imagine that you falsely predict genetic variation fairly commonly when it's actually just sequencing error. And that's a problem, especially when you're talking about rare events like mutations in a population of cancer cells. If the frequency of those mutations is roughly the same as the frequency of the error or of errors, the error rate, problems happen quite frequently. So for instance, if your error rate is 1% and the mutation rate is 1%, naively, that means that 50% of the things that you think are a mutation are in fact a sequencing error. Okay, so back to the point. We get this big hideous text file. It's literally a text file, which you can scroll through if you were very bored on the Unix command line. And our goal, and this is the focus of today's lecture, is the crucial first step in doing anything with these data is to align or map and align these DNA sequences, billions of them, to a reference genome. If you're doing RNA-seq, that's required to get a first count of cDNA molecules to measure expression. If you're finding genetic variation, that is the first step in trying to figure out where the DNA from the individual that you sequenced differs from the DNA that is in the reference genome. And those would be potential sources of genetic variation. you're doing ChIP-seq, it's the same thing. ATAC-seq, there's so many different -seqs now, I can't even name them, but row-seq and clip-seq, and I don't know, you guys are probably doing ones I've never even heard of, but they all essentially start with alignment and mapping to a reference genome. So we can count things because that count is the proxy for the biological or cellular phenomenon that we're trying to measure with a sequencing assay. Right. So I think conceptually, mapping and alignment to a reference genome is easy. It's easy to be able to think about, okay, I take this sequence here, ACCCC, TGG, and I figure out where it belongs in the reference genome. Great. I do it for the next sequence and so on and so forth. What I'm going to talk about is really what I'm going to focus on today is why it's not so easy. All right. And there's a laundry list of reasons. The first is the human genome, but this is true really for any model organism genome. Zebrafish genomes, mouse genomes, human genomes, Drosophila genomes, they're all relatively large and they're all complex. And what I mean by complex is that they're repetitive. There's lots of the same nucleotide sequence that's littered throughout those genomes. And that's for many reasons. Most genomes have evolved in part because of retrotransposition. So sequences are copied and pasted into different parts of the genome through an enzyme called retrotransposase, which also can insert other exogenous sequences like mRNAs and things like that. So we have this big, complex, repetitive genome. And the point of saying that is that that makes it difficult to figure out unambiguously where a given small DNA fragment that comes off of these sequencing machines and is in a FASTQ file makes it difficult where to place it, figure out where to place it. Okay, so the genomes are big, scary places. And then the other problem is we have to, when we do these sequencing assays now, we're getting billions of DNA molecules out of these runs, sometimes millions for RNA-seq, but if you're doing whole genome sequencing of a human genome, you're talking literally billions of reads. So we need to be able to do this fast. Some of the early sequence aligners, it would have taken weeks just to align a single human, the DNA for a single human genome sequence. In the context of precision medicine, if it takes weeks or months to align the data, it's not terribly clinically relevant if you're trying to find genetic variants, for instance, that might explain why an infant is sick. So we need to be able to do this fast in the face of the huge complexity of big, complex genomes like the human genome. I think one of the central problems also is that sequencing technologies make mistakes. And I say frequently, yes, the error rates now are less than 1%. They're even probably less than 0.1%. So, barely more than 1 out of 1,000 nucleotides are wrong. But when you're talking about billions and billions of sequences that each have 150 nucleotides, you're talking about 150 billion to 300 billion nucleotides, and with an error rate of 0.1%, that's still a lot of errors. So we really want to make our best effort to get accurate alignments, and the accurate alignments take time, but what I'm trying to emphasize is that it's really worth it, because if you take shortcuts and don't align your sequences with the best strategy for RNA-seq or for ChIP-seq or for DNA sequencing, it can lead to artifacts in the interpretation of your data. And that's a waste of time, effectively. Let's spend the time up front to do the alignment right, so that we don't chase red herrings in the analysis. And the last thing I want to emphasize is that there are specialized alignment strategies depending upon the experimental context. For instance, we typically, the most widely used aligner for DNA sequencing, for exome or genome sequencing, is something called BWA, the Burroughs-Wheeler aligner. However, for RNA-seq, the Burroughs-Wheeler aligner is not terribly widely used. Typically, it's STAR or HiSat2, I think, is the other sort of 800-pound gorilla of RNA-seq alignment. And the reason for that is those different aligners, BWA versus STAR and HiSat, are expecting different alignment patterns. Namely, in the case of DNA-seq, you don't expect to have sequences that span exon-exon junctions where introns are removed. But for RNA-seq, since we know we're dealing with mRNAs, we can have lots of sequences that get split across multiple exons because of splicing. All right, so spent a lot of time going through that, but the motivation here is that alignment really is central to all genomic research. We start with this FASTQ format, which we'll talk about. We have to align to a genome. We got to do some quality control. And then, so everything that's in black or gray here is basically, it's relevant to every type of genomic assay: RNA-seq, ChIP-seq, CLIP-seq, DNA-seq, single cell, RNA-seq, ATAC-seq, whatever. Where things start to differ is essentially what you do with those alignments, how you count things, how you find peaks in the case of transcription factor binding. Are you looking for mutations? Are you looking for inherited variants? Are you trying to find differential expression? Are you looking for alternative splicing, et cetera, et cetera? And there's all sorts of specialized software tools that you end up using in this green phase, which is like post-alignment. What do you do with the data? And then, phase 2 through maybe even 1,000, not just 100, is this iterative process of, okay, I've aligned the sequences, I've run my software, I do some analysis, I try to interpret the data, and some things may look interesting, but I have to be skeptical. I might think that this looks interesting, but I'm hopefully aware that there might be technical or experimental biases that lead, or artifacts that lead to that seemingly interesting observation. So I have to go back and make sure, do some quality control, rule out technical artifacts that masquerade as biological signal. And that can often, that phase for some PhD thesis projects can be 3 years, literally, just redoing experiments, making sure that the signal is reproducible, figuring out how to develop new methods to tease out what's signal and what's noise. All right. So if we have these FASTQ files that you haven't seen yet, but just imagine in your head a text file of billions and billions of DNA sequences, our challenge for mapping and alignment is to find a home in a reference genome for every single one of those reads. And when I say read, that's jargon for a DNA sequence in a FASTQ file. So finding a home for every read in the file and a place in the reference genome where it could align is called mapping. So essentially you're mapping or projecting the sequence to potential locations in the genome. And I say locations because of the fact that the human genome is repetitive. Some sequences, if it's say a line element or one of the richer transposon families, if it comes from a line element, there could be hundreds, if not thousands, of potential locations in the reference genome that look very similar to that DNA sequence. Then, for each of the possible mapping locations in the reference genome, a sequence aligner must get the exact alignment right. And by alignment, what I mean is the precise nucleotide by nucleotide registration of the DNA sequence that's in the read that came out of the FASTQ file versus the reference genome. We'll get into the details of that. And given what I told you before, we have to use the right tool, the right aligner for the right experiment. And this is usually talking to colleagues, going to read previous papers that have published on ATAC-seq or RNA-seq, and seeing what methods they used and what justifications they gave for using that tool. All right, so I mentioned the fact that the human genome is comprised of repeats. We've seen this slide before. It's about, this is an early estimate. I think this might be from around 2010, where the estimate was that about 45% of the human genome was retrotransposon or transposons of some sort. Retrotransposons here, and then DNA transposons are barely 3%. So from a big picture, this is basically saying essentially half the genome is repetitive in some way or another. So on average, half of the sequences that we get out of a FASTQ file from a sequencing experiment are going to pose some difficulty in terms of figuring out where exactly that sequence maps to or belongs to in the reference genome. And I've mentioned this before, but these are retrotransposons are really copy paste. So basically they get transcribed, they get reverse transcribed, and they get inserted by retrotransposases into a new location. So this is sort of the donor site in the genome, and they can, if they get reverse transcribed in the nucleus, then they can be inserted into new target sites in the genome. In contrast, DNA transposons are actually cut and paste. They are removed from their donor locus and placed into the target locus. So retrotransposons are what lead to the growth or the expansion in a genome size, because essentially it's copying and pasting. While they're still, when retrotransposons are active in a species, that genome is sort of in a tug of war of growing, but also there seems to be a limit on the size of a chromosome. So most, and for instance, the number of genes. I think one of the really interesting, to me, one of the very interesting things in genome evolution is why there seems to be an upper limit on the number of genes. There doesn't seem to be much constraint on the size of a genome. Genomes can span orders of magnitude, but if you think across even model organisms, the sort of the mode gene number is something around 18 to 25,000. Why is that when there's so much plasticity in the size of the human of genomes themselves? If you can answer that and figure that out, that's a pretty huge advance. Okay, so half of the genome is comprised of repeats. Really, what does this look like? Well, here's just a little tidbit, snippet of human chromosome 1. You can see that there's these low order repeats. This is at the in the telomere. So this is a telomeric repeat. So we get repeats that are on this order where it's short little 6 nucleotides, or 6mers as they're known. So that's one order of repeat. Those are so-called simple repeats or microsatellite repeats. And then there's these higher order repeats that come from transposons. So line elements, oh gosh, a line element is about 6,000 base pairs. If I remember right, if you know this, if this is your area of expertise, correct me, but I'm pretty sure they're about 6 kb. When they copy and paste, you get repeats that look like these red sequences, but the sequences are now 6,000 base pairs, and they're littered throughout the genome. So the problem is, this is my analogy to what the human reference genome looks like. It is, and I'm thinking of this as a jigsaw puzzle. Probably all of us in the first couple months of COVID did a lot of jigsaw puzzles, and we got pretty advanced in my house, and we started doing some really challenging ones like this, where it's difficult if you get a small DNA sequence like up here, a little snippet of DNA by analogy that is the equivalent of a small jigsaw puzzle piece. It's difficult to know whether that sequence belongs here or here or here, or even down here in the water. And it's these subtle differences like ripples or edges of clouds or the exact pixel composition that allow us to figure out, have some greater certainty that a particular sequence belongs here versus here versus here. But I think what this analogy does is give you a sense of the computational challenges that aligners are having to face when trying to figure out where, say, 150 nucleotide sequences that we get out of an Illumina sequencer belong in the reference genome, given that we have repeats that are on the order of 6 kb or 500 nucleotides. And that difficulty is what underlies the excitement for long-read sequencing technologies that you may have heard of, like PacBio, Pacific Biosciences, and Oxford Nanopore, because they're longer, much longer sequences. So that's like taking this complex jigsaw puzzle, and instead of doing this, where we have small little pieces that come out of an Illumina machine, we get, the kind of puzzles that we did when we were kids, where it's broken up into just like 40 pieces, and it's kind of easy. There's a lot of information in each of the individual large pieces that help us figure out where the overlaps between these pieces are. But when the pieces or the DNA sequences are very short, it's difficult to disambiguate where a given sequence belongs. Okay. Now, let's talk about how this actually works, how sequences are mapped or aligned. So here's the process. We've got a bunch of DNA that we put into a sequencer. I've actually never literally done this, but you typically isolate the DNA and then hand it off to a sequencing core or ship it off to a company. Then somebody there puts it into one of these sequencing devices that cost, like, this one's probably about $500,000, the HiSeq 2000. And then 24 hours go by through a process I'll explain next time. The sequences for the DNA that was put in are inferred, and you get this FASTQ file out. The aligner has to take each of those sequences and figure out where they belong. Now, computers, as you've learned with grep, are really, really good at finding exact matches. So grep is great at that. Google, if you search, or other search engines, what are they? I guess the only one I know about that people make fun of is Bing. But there's probably other ones. Google, they're really good at just scanning and indexing the whole internet and finding exact matches. And so if the best case scenario for DNA sequencing for mapping is if the sequencing technology makes no mistakes. So if the DNA that goes in is exactly the DNA that comes out, well, this aligner can, you could imagine it's just like grepping for these sequences. It finds places in the genome, highlighted in blue, where those sequences match exactly. But as I mentioned, errors happen. So let's imagine this is the DNA that goes in, ATTC, but in synthesizing, integrating this nucleotide into the sequence that's being sequenced on the device, it makes a mistake, and it falsely guesses that the fourth nucleotide incorporated was actually a T and not a C. So now we've got a problem. Now the aligner has to explore many other possibilities. So it's got this ATTC possible match. I underline the difference or the mismatch between the sequence that it's given and the potential home in the reference genome. There's another place where it's ATTG, and then the rest of the nucleotides match. And then there's a third one, which is ATTA. And there's actually no places in this toy reference genome where the sequence ATTT matches exactly. So this is called fuzzy matching. And the whole point here is the more errors there are, sequencing errors, the more work that the aligner has to do, because it has to explore many possible other fuzzy matches or close matches. And this is kind of like me when I Google something and I 99 times out of 100 don't type the whole thing properly without an error, and it'll usually say, well, did you actually mean this? And I usually click yes. and it goes out and searches, but it had to detect the fact that what I typed didn't quite match a normal search term. Okay, so one thing I want to emphasize here is that not only does the aligner have to do more work when there are errors, that's a difference or a mismatch, but also if there's genetic variation. If let's say this actually isn't a sequencing error, let's say it was a T up here as well, and my DNA, I inherited this T allele from my mom. And so that T allele is not in the reference genome. The aligner has to do more work to find this difference, that true difference that exists in my DNA versus the reference genome. So aligners have to take into account the possibility that differences between DNA that it's being asked to align and the reference genome come from either A, sequencing error, or B, true genetic variation. And that challenge is variable. The degree to which that's difficult is variable from species to species. So for instance, any two humans, we'll talk about this more later, but any two humans, if we say sample my chromosome 1, my paternal chromosome 1, and compare it to my maternal chromosome 1, there will be a difference on average, like 1 out of every 756, or maybe it's 1 out of 762, I can't remember, nucleotides. So that's what's known as sort of the heterozygosity, or the degree of genetic diversity in the human genome. 1 out of every 762 positions differ on average. So doesn't seem like that much work for the aligner. It's like, it's pretty infrequent true genetic variation difference. But when you get down to dealing with like diverse strains of Drosophila, you're talking about nucleotide diversity of about 1 out of 100 nucleotides. And if every sequencing read that you get is 150 nucleotides, on average, every single one of those reads is going to have one difference. If you're sequencing a wild strain of Drosophila, it's going to have one difference on average as compared to the Drosophila reference genome. Okay. So mapping versus alignment. What I just showed you is mapping. So finding potential locations for a given sequence in the reference genome. So ideally, we want to do this fast and find the n best possible loci to which a sequence could be aligned. So good candidate places to place the jigsaw puzzle piece of the short DNA sequence on the reference genome, or the box that you're looking at. Then alignment is for each of those n loci that are potential mapping locations. You want to then do a second step, which is that nucleotide by nucleotide registration, which would allow us to realize that there's a difference between this sequence this sequence that we're trying to align and this place in the reference genome at exactly that nucleotide. And that nucleotide by nucleotide registration or alignment is what's required for us to be able to detect genetic variation. Hold on a second. Not sure why I would get calls here at this time. That allows us to find mutations in cancer genomes and in genetic variation, and that's inherited from our mother and father. Okay, so now we're going to go through a toy example of how mapping actually works. There are two main strategies for mapping, finding potential loci in a reference genome to which a sequence can align. One is called hash-based mapping, and another one is called the Burrows-Wheeler transform. We're not going to talk about the Burrows-Wheeler transform because it's much deeper in computer science, but I think the one thing that I'll say is the hash-based mapping approach that I'll talk about now is basically the same concept as what's used for Burrows-Wheeler aligners. It's just that the hash, which I'll explain in a second, is just a far more sophisticated data structure that these aligners, the Burroughs-Wheeler aligner, BWA, and you've probably heard of bow tie. The B and the W in bow tie stand for Burroughs-Wheeler. And just a quick historical aside, the Burroughs-Wheeler transform was figured out by Burroughs and Wheeler, and it underlies a tool in Unix and a compression algorithm called bzip. And that's this fancy strategy for figuring out how common patterns in a text file and how to rearrange the file so that it's much more compressible. Okay, so let's imagine we're lucky and we work with a very simple model organism that just has this toy 16 base pair genome right here. The way hash-based mapping works, step one is to actually take the reference genome itself. So this toy genome or the human reference genome or the mouse reference genome, and we so-called hash it. And hashing, it works as follows. What we do is we choose a hash size, and the typical variable to represent a hash size, or a k-mer, you've probably heard of k-mers before, is we typically choose an odd number, and that's to avoid try to avoid palindromes as much as possible. It doesn't avoid all. But if k equals 3 in this case, we literally start the beginning of the genome and we break up. We find the k-mer starting at position 1. In this case, k is equal to 3. So the first one is CAT. And what we do is we're building up a table that the algorithm uses. And this table is the index for the genome. And we'll explain how all this works. But CAT starts at genome position 1. Then we slide to the next nucleotide, and we get the next k-mer, ATG. It's at position 2. TGG is at position 3. GGT 4, GTC, so on. But when we get to CAT again, we don't add a new entry to this table. We track the fact that we've seen CAT before. The first time we saw it was at position 1, but we've seen it again now at position 7. So we're building up a table of all the distinct k-mers that exist in the genome and the list of the positions. So for DNA sequences, how many possible k-mers of length 3 are there? Anyone have an idea? How many nucleotides are there? Let's ignore ends. How many possible nucleotides are there? There's 4. So the number of possible k-mers is 4 to the 3rd. So there's 64 possible 3-mers. Okay, so we go on and on. We march through the whole genome, and we end up with a list of all the k-mers that we observed, and then a list of the positions at which that k-mer was observed. Okay, this is our complete hash index, also called a k-mer index of our toy genome. And for simplicity, I just did this on the forward strand, the reference strand, but you could also imagine doing this on the reverse complement strand and tracking all those k-mers as well, because we can get DNA sequences that are on one strand versus the other, right? Okay, so that's our index. This is step 1. This doesn't align DNA sequences. We haven't even looked at DNA sequences yet from a FASTQ file. This is just basically building a lookup table in the reference genome. And so this is very much, it's called an index because this is very much like an index at the back of a book. It will tell you where certain words, the list of all the page numbers where words are. So that, I think, conceptually is a much faster strategy. If I want to find all the occurrences of the word Aaron in a book, I would much rather refer to an index, look up Aaron in the index and find all the page numbers, then start at page 1 and turn the page and read the whole book and, get a pen out. If someone has already pre-computed that for me, let's use it. It speeds things up. And, while that may seem kind of simple for this 16 nucleotide genome, when the genome is 3.1 billion base pairs, trust me, having that index to do this quickly is very beneficial. So how do we use the index? Well, we have to know when an aligner goes to align a sequencing read using this hash-based index, it has to know what the hash size was when building the index. So I told you that we used hash size 3, or I showed you that we used hash size 3. So when we want to align this sequence, we have to break up the sequence using the exact same strategy that we use to break up the reference genome. So let's take this read TGG TCA, and our job right now is to map this read to the toy genome. And so we want to find possible loci to which that read can align. So the first thing we do is we start the first nucleotide and we get the first k-mer, 3-mer. And what we do is we don't look that up in the full genome, we look it up in the index. Because remember, this genome could be 3.1 billion nucleotides, but this k-mer index is going to be much smaller. We've broken up into smaller chunks that we can digest a bit better. So we look up TGG in this hash table and we see, well, there's 2 possible matches. It could start at position 3, 1, 2, 3. Yep, look, TGG. And it could start at position 10. There it is again. So that gives us 2 candidates, but there's more information to winnow this down a little bit in the read. We can then look at the second 3-mer. And for simplicity, what I'm going to do now is just skip to the next k-mer that starts at TCA. I'm not going to do it from each position. Then what I can see is, TCA starts at position 6. That's interesting, because if I have a candidate that starts at position 3 for the first k-mer, and I know that my length k is equal to 3, and I see this other k-mer TCA starts at position 6, well, position 6 is 3 nucleotides away from position 3. So that tells me automatically that there's an exact match for this whole sequence, TGG, TCA, starting at position 3, because the offsets of the positions of these hash matches. So we can just say, all right, TGG belongs to 3, and then the second k-mer that I know is shifted 3 nucleotides over from the first one, starts at position 6. Well, 3 and 6 are 3 nucleotides apart. Great. I didn't have to do any work. I don't have to do any alignment. And I've used this hash table to quickly infer, essentially with effectively geometry offsets here, that this is an exact match. So if it's an exact match like this, I don't have to align anything. I don't have to do a more complicated algorithm that I'll talk about later in this lecture. I just know it's an exact match. So I am done with this read. Okay, so that was a bit easy because the read and the reference exactly matched. But I told you that the hard work in DNA sequence alignment is when there's an error or a genetic variant in the read. So let's see what that looks like. So in this case, now we have an error. TGG still has an exact match, but this TCT doesn't have an exact match in the reference genome. So we get TGG, we get that positions 3 and 10, and then we've got, TCT doesn't match. This is a case where we would actually have to test. We'd have to say, all right, I'm going to take this whole sequence. I'm going to start the TGG at position 3, and I'm going to see how well the second part of my sequence, TCT, matches 3 nucleotides after this TGG match in the reference genome. And that process, comparing T to T, C to C, and T to A, is alignment. We're basically scoring that alignment. Is it a good alignment? Well, 5 out of 6 nucleotides matched. Sure, that's pretty good. Now let's compare that to the other option where we start this TGG sequence at position 10. Well, then we're looking at TGG, TGG followed by TTC. Well, in that case, just the first T matches, and then the C doesn't match T, and the T doesn't match C. So that's 2 differences, versus if we started the alignment at position 3, we'd only have 1 difference. So if we were thinking about the ranking of these, we would rank the alignment starting at position 3 as more confident of the true location of this DNA sequence in the reference genome than the second alignment starting at position 10. Does that make sense? Okay. Right. So I took you through a really simple scenario where we have this sad little toy genome with 16 nucleotides, and we have k equal 3 because that made for a nice, simple to explain slide. But one of the challenges in using a hash-based aligner is what's the size of k that's best to use to facilitate mapping to the reference genome. So I guess let's think through this. I mean, what is a good choice of k? Is k equal 1 a good choice for mapping reads to the reference genome? If our whole idea is to build up an index that's useful for finding ideally a small subset of loci in the genome to which a sequence could align. Would k equal to 1 work well?
No.
Okay, why?
Because then you're just matching singular base pairs. You're not matching like chunks or strings of them.
Exactly. So if we had k equal 1, there's going to be 4 entries in our hash table, A, C, G, and T. And roughly, well, GC content, I forget. Well, let's just keep it simple. There's 3.1 billion positions in the genome. Each of those A, C, G, and T will have roughly 3.1 billion divided by 4 lists of locations to which that could align. So it's not very informative. That's right. Similarly, k equal 2, not terribly informative. Now we got 16 possible entries in that hash table. And so 3.1 billion divided by 16, better, but not great. When k equals 3, we have 64 possible 64 possible locations. K equal 4, I think, is 256. K equal 5 is maybe 1024, something like that. So one of the problems is figuring out what the optimal choice of k is, depending upon the composition of the reference genome. And on the other side, we don't want the k to be too big. What if k is equal to 3.1 billion? Well, we have 1 entry, the whole genome, with 1 number in it, starts at position 1. Not very useful. So there's this sweet spot in between, which is, having k being large enough where it's informative, but not so large that it transitions quickly to being uninformative in terms of finding possible locations. Now, I can just jump ahead and tell you that most hash-based aligners use 31 mers, k equal 31. So it shreds up the genome into 31 base pair, sometimes 27 or 31 base pair entries. And the reason for that has to do with basically the composition of the human genome. Okay, so Jordan already destroyed this idea of k equal 1. No, that's a terrible idea. 4 to the 3rd, we didn't like that. K equal 10 is actually an interesting, so in the early days of alignment, and so the other trade-off here is when k gets large, you start to use more and more computer memory to store that index in computer memory. So there's also this trade-off of, we might like the k to be a little bit larger because it might be more informative, but we don't have enough memory on, say, my laptop to do the alignment, because you can't actually load that index file up into RAM to do the alignment really fast. And that was a big problem. My first laptop in grad school had 128 megabytes of RAM, and was far less than an index for K equal 31. So one of the this paper that I'm referring below, you don't have to read this, but if you're interested in this K composition or K-mer composition of human genomes, there's actually a lot of literature on this. But if K equals 10, that means there's 4 to the 10th possibilities, which is this number, 1 million and change. It turns out that every one of those 1,048,576 K-mers, 10-mers, those are the theoretically possible K-mers for K equal 10. Every one of those exists at least once in the human genome. But you can imagine that when K gets large enough, let's do K equal 31. K equal 31 is a stinking huge number. It's like 4 to the 18th. 4 times 10 to the 18th. That's way bigger than the human genome. So only a subset of the possible 31 mers exist in the human genome. All right, so let's jump out here. So what this comes from this other paper that I'm referencing here at the bottom, is what we're looking at is K-mer length. That's way down here. I think the lowest they go to is maybe 10. And what they're looking at is, given that K-mer length, what fraction of the K-mers in a particular genome are unique? That is, they occur once and only once. So an extreme case is if K equals 3.1 billion, well, that K-mer exists once in the human genome. But if K equal 10, I told you that every one of those occurs at least once. The question, what this is plotting on the y-axis, is what fraction of those 10-mers occur once and only once. And that's a proxy or an estimate of how unique or how repetitive a given genome is. So for instance, chicken genome is about a gigabase pair. It is in red here, and you can see that as K increases in size, chicken and dog, which is in orange here, the fraction of K-mers that are unique in those genomes increases very quickly. In contrast, for fruit fly, it takes a bigger K-mer length to get a higher proportion of K-mers that are unique in the Drosophila genome. And that's, if you're a Drosophila geneticist, you can help me out here, but there's all sorts of retrotransposons in the Drosophila genome that are still active. One I remember is Mariner, and that's about the extent of my Drosophila retrotransposon knowledge, but they're super active. So there's lots of repetitive sequence in the Drosophila genome. Therefore, it makes sense that it takes a higher K-mer size to get a higher proportion of unique sequence in the Drosophila genome. And human is, sort of between the two. It takes a very large K-mer size. So let's go way out here to 1,000. For human, when you go out to K equal 1,000, it's still only like, let's see, 92.5, 95, 97, about 98% of K equal 1,000 K-mers are unique. So there's still, 2% of 1,000 base pair sequences occur more than once in the human genome. And it's this tail way out here at large K-mer size that makes sequence alignment hard when the sequences are short. Okay. Right. So that sets up, so we have this problem where K-mers are repetitive. And so we have to have some sense of the uncertainty about an aligner's choice the aligner says, all right, I've used my K-mer index. I see all these locations. I've done fuzzy matching and tried to figure this out. And you know what? I chose this location because it seems best. But I need to tell you that there were other possible locations. I need to reflect that uncertainty about my choice versus these other choices in a score. And this leads to something called mapping quality, or MAPQ, as we'll talk about in the BAM format. And this MAPQ reflects the probability that the given sequence should be mapped here and only here. So if there's only one locus, the mapping quality should be very high. So the higher the score, the more certain that the aligner was if the sequence belongs here and only here. If the mapping quality is low, it's reflecting the fact that there were lots of other choices, and I kind of just flipped a coin and chose one. All right, so this, I'm going to try to use my whiteboard briefly here. Let me, if I write like that big, can you see it? Or is it terrible?
Terrible.
Terrible. All right, skip that. Thank you for the feedback. I'll try to explain this in words. This is where whiteboards are so great. All right. So the way this math works is it's based on something called the Phred scale, PHRED. The PH stands for Phil Green. He's the guy who came up with it. He's a professor at University of Washington. So it's somewhat eponymously named. So basically what we're doing is you're taking, let's start with the parentheses. What's in the parentheses? The probability that the mapping location are reported to you is wrong. So let's say the probability that the mapping location is wrong is 0.1. So there's a 10% chance that I'm wrong. What this does is it multiplies that probability. It takes the base 10 log of that probability. So if the probability is 0.1, that means the base 10 probability of that is -1 because you're just shifting the decimal place one to the left. And then what the Phred scale does is it multiplies that by -10. So it flips the negative sign. So we get higher numbers and multiplies by 10. So a mapping quality of 10 in Phred scale means that the base 10 log of that probability was -1, which means that the probability of that the mapping location is wrong is 0.1. Similarly, if the mapping quality is 20, it works out that the probability of the mapping location is wrong is 0.01. So I think of this as 2 decimal places shifted from a probability of 1. 3 decimal places shifted. 1 in 1,000 chance that I'm wrong leads to a mapping quality of 40. 1 in 10,000 chance that the mapping location is wrong gives a mapping quality of 40. Hopefully I didn't say 40 for the other one. But it leads to this kind of intuitive factor of multiples of 10 rise in the mapping quality reflect multiples of 10 decrease in the probability that the mapping location is wrong. And we'll come back to the Phred scale in the case of sequencing qualities next week. But essentially, if you don't want to memorize this equation, you don't have to. But essentially, the higher the quality, the more confident in the mapping location. And just try to remember that these are factors of 10 reduction. As it goes up, it's factors of 10 reduction in the probability that the mapping location is wrong. And this really comes into play with, if we have a sequence, a DNA sequence that is fully contained within, represents a line element, which is a retrotransposon. And let's say there's 50 entries in the reference genome for that, the mapping quality should be low. But if the DNA came from sort of unique sequence on the right side of this 3D fancy pie chart, then we would say that we would infer that the mapping quality should be high. There's more confidence that this is the one and only one location that sequence should align. I'm not going to belabor this, but basically the point here is that here's mapping quality scores on the x-axis, referring to very uncertain alignments, mapping quality 0. That basically means if it's mapping quality 0, that means the aligner is certain it's wrong. It's just guessing one. It's choosing one location, but there's thousands of other equally good. Versus, way out here, we're talking about, map quality 40 means that there's a 1 in 10,000 chance that the location was wrong. What I'm showing here is just the scores, the mapping quality scores that come from two different aligners. They're Burroughs-Wheeler aligners, BWA and Bowtie. And you can see that they have a different distribution of mapping quality scores that they assign. And it's basically because, there is no truth to this. It is the aligners are trying to estimate the probability that the mapping location they report to is wrong. It's not an unequivocal truth. And that you can tell here that they use different estimators. And therefore the probability or the density distribution of these mapping quality scores differs between the two. I present this just so you're aware of it. I would say today, fast forward to right now, if you look at mapping quality scores, if you just focus on alignments with a mapping quality score of 20 and higher, those are typically fairly confident alignments, and the aligners have sort of converged on better assessments of mapping quality. So 20, sometimes mapping quality of 30, 1 in 1,000 probability that's wrong is a good rule of thumb for identifying high confidence alignments or mappings. Okay, so now we're going to transition from mapping, where we've placed sequences to possible locations in the reference genome. And we're going to talk about now alignment. But before we can talk about alignment, we need to talk about a concept called edit distance. And that's a fancy way of saying just how many changes, edits, must be made to one word in order to make it match another word. And word can be literally words in the English language or any language, or DNA words, k-mers. So let's just take an example. If you compare curled versus hurled, there's an edit distance of 1 between these, because you can either change the C to an H and they match, or the H to a C and they match, yeah? Okay, so more complicated example, slightly more complicated is shot versus short. The edit distance is still 1, but the edit type is different. Now we would either delete the R from short to make it shot, or we would insert an R in shot to make it short. And okay, so now by analogy, what does it look like for a more complicated scenario like this, where we've got two sequences that have some similarity, but a lot of dissimilarity? Well, one solution would be to align the two sequences with the following 5 edits. We could essentially delete the T from this sequence. I'm sorry, we could insert a gap between these two sequences to accommodate the T. The G's match, and then we have to insert an extra G, and then we get 2 T's to match, and then we have to accommodate this extra G in the one sequence, and we have AC in matches, and then we have 2 mismatches at the end. So in total, we have 1, 2, 3, 4, 5 edits. Another solution would be to do the following. We incur a mismatch at the beginning, we align GTT, and we have a gap, and then we have mismatches at the end. So in this case, there's an edit distance of 4. So arguably, this second solution is better than the first, because the number of differences between them, the number of accommodations we had to make is less. So this introduces the question of what is the correct alignment? And I think the main point I want to say for the second part of the lecture, I'll just say it up front, is there is no correct alignment. There is no biologically correct alignment between sequences. It's an unknown entity. There is a computationally optimal alignment given a set of rules that an aligner follows. And I'm going to talk about something called the dynamic programming algorithm, which underlies two things you may have heard of before: the Smith-Waterman aligner and the Needleman-Wunsch aligner. These are advances that were made in the 70s and 80s to basically come up with optimal solutions for this type of problem. Sorry for the noise. Someone apparently is sledgehammering a wall right next to me. It's really fun. So what we're going to get into is, how do we use a rubric, a rule system, to find the optimal alignment? And the other key thing I want to emphasize is that the optimal alignment is different depending upon how you reward matches or mismatches or gaps in these sequences. Okay. So the two main papers that came out in this area were one in 1970 from Saul Needleman and Christian Wunsch. That's the so-called Needleman-Wunsch algorithm. And then 11 years later, T.F. Smith and Michael Waterman came up with another approach called the Smith-Waterman algorithm. We're going to focus on the Smith-Waterman alignment approach today and explain how it works. Okay, because this is the more widely used approach for aligning short DNA sequences in the context of RNA-seq and ChIP-seq and DNA sequencing. Okay. All right, I have 15 minutes to get through this, and I think we can do that no problem. So what we do is we take the two sequences in question. So we're going back to these two sequences here, and we place them on two axes of a matrix. Okay. And what we do is we insert an extra row and column. So that's this offset here. And that's basically so we can follow the algorithm that Smith and Waterman outlined. Before we get into the details, the reason for adding this extra row and column is so that we can align any base in one sequence to any of the other positions in the other sequence. Sorry, my block. So we'll see why that's useful in just a second. Now, the second part of the dynamic programming algorithm, dynamic programming is the fact that, as we'll see, there's many possible paths that we can follow. So it's a bit of a choose your own adventure. And it's dynamic in the sense that once we populate this matrix with scores, I'll show you what I mean in a second, we can dynamically figure out the best path through those scores. Now, the optimal alignment and the way we populate this matrix is governed by a rubric or a scoring scheme. So what Smith and the advance that Smith and Waterman gave us is, if you define rules for how you want to reward cases where nucleotides match or exact, or when they differ, they're mismatches, or rules when we have to insert or insert a sequence, a nucleotide in one sequence versus the other, introduce a gap in the other sequence, how we want to penalize those things. The algorithm that I'm going to show you will give you an optimal alignment given that scoring scheme. That does not mean that it is the biologically most relevant alignment, but it turns out that through many decades of work subsequent to Smith and Waterman's efforts, there've been some fairly well-established rewarding schemes for rewarding a match and penalizing mismatches and penalizing gaps that seem to, well, we don't know them to be the biologically optimal. All signs point to these are generating alignments that really model mutation dynamics and the way genomes evolve, and so are a pretty good proxy for the biologically most optimal alignment between two sequences. And what I mean by that is, if we see differences, do we really, between two alignments, can we think about them as true genetic differences between the sequences, or is it just artifacts of the scoring scheme? We want the former, not the latter. All right, so we build this matrix where we add the extra row and column. And what we do is we first initialize that row and column to a score of 0. Okay, I'm just following procedure here. We'll come back to why this is in a little bit. So that's how we start. Then we start in the upper left-hand corner of the matrix. And what we do is we move to the first unpopulated, first unscored cell in this matrix. And when we look at that, we compare these two nucleotides. In this case, we see that it's a mismatch. So the scoring scheme says if it's a mismatch, we need to subtract 3 from the score of the cell from which we came. So we move from this cell to this cell. So the rules tell us we should subtract 3 and the score should be -3 here. But wait, there's a catch. The other catch with the Smith-Waterman algorithm is that the score can never become negative. If it hits at 0 and you have a -3, you just keep it at 0. And we'll see why in a little bit. Okay, so that's why this score now, we moved from this cell, its score was 0. We see a mismatch, but we keep it at 0 because we can't go below 0. Now, if we go in this direction, we go moving from this cell to this cell, and this cell to this cell, and this cell to this cell, we see G versus G. That's a match. So we increase the score by 3. Okay. And we move on and we look at every cell, and you're essentially looking at the previous cell, what the score was, and what the nucleotides are at the current cell, and we update the score accordingly if it's a mismatch or a match or a gap. So let me focus in on this cell as an example. So right out here, we're on diagonal. The score at this cell is 9. If we move this way, that would be the introduction of a gap in the alignment. And so that allowed that because we've gone off diagonal. So if you go off diagonal, essentially you are introducing a gap in one sequence versus the other. Oh, and this is my note to fix a mistake that I found last year that I fixed. Right. So we introduce a gap, so we have to penalize this previous score of 9 to 7 when we move in this direction. Same this way, we've moved off diagonal in the other direction, so now we're introducing a gap. And if we go this way, G versus A is a mismatch, so we have to subtract the score by 3, and now the current score in this cell would be 6. Okay, I know this is probably a little boring and befuddling. We'll see why this is useful in just a minute. We follow this procedure, and the rule is you have to populate every cell in the entire matrix that you built to compare these two sequences. So you can imagine, this is a 1, 2, 3, 4, 5, let's see, 9 by 10 matrix, something like that. So there's what, 90 cells that we have to fill. Computers are fast, but if we have to fill this cell by basically, following this algorithm for comparing every one of billions of sequence to a reference genome, this is a lot of work actually. And this is only step 1. We're using this scoring scheme to populate this whole matrix. Now, how do we use this matrix to dynamically, hence the dynamic programming algorithm, dynamically find the optimal alignment? Well, the next step is we find the cell in the matrix that has the highest score. So this is where the score of the alignment is maximized between the two sequences. And we essentially work backwards from there. So we know at that cell, it was a C versus a C. So that's how we basically form the alignment from the right now. We're working backwards from right to left. And then we find, going from this cell, this is our start of the alignment. We find the cell coming off of that. This is the dynamic part. We're choosing our best adventure here, where the score remains highest if we move this way. Okay, and that makes sense because it's a match between A and A, and that's what we put in. Oops. Now, what we do is this introduces a gap between the G and the A. So we have to, coming from this direction. And so when we work all the way back, following this path, what's the best score all the way back and introducing the right operation in the alignment, depending upon whether we went off diagonal or stayed on diagonal. We end when we get to score equals 0. Okay, so the last nucleotide that we include is G versus G, so that's here. So the question is, why do we not include this last bit when we get to 0? Well, that's the other hidden trick in the algorithm, is when we know we get to score equals 0, then that's where the alignment should end. So the rules of the Smith-Waterman alignment, and this is why it's called a local alignment, is it never starts or ends with mismatches. You're providing a local alignment where the alignment starts and ends with matches, and there might be mismatches and gaps in between. So we exclude this G versus T, that could be right here, but it's omitted. And we're also excluding this T versus G and A versus G at the end. So local alignments, the optimal local alignment need not include every nucleotide in the 2 sequences. And that's where the Smith-Waterman alignment, which is local, as I just explained, differs from that Needleman-Wunsch alignment. The Needleman-Wunsch alignment, which came out in 1970, is global in that every nucleotide in the sequence must be used. But at the consequence of probably introducing lots of mismatches and gaps that are required to use every nucleotide. But if we were looking for genetic variation using those kind of alignments, we would probably find a lot of spurious things that look like genetic differences, but are actually just the consequence of being forced by the algorithm to align everything, even if it's not, even if it introduces mismatches that masquerade as genetic variants. So it's that reason that the Smith-Waterman alignment, which is local and doesn't start or end with mismatches, is used to find genetic variation, because it's registered or tracked the nucleotides that are sort of at the core of the common subsequences between the 2 sequences aligned. So you're not going to be tested, and you'll probably never have to write the Smith-Waterman algorithm. You may never have to think about it again. But I think it's a term, dynamic programming and Smith-Waterman and Needleman-Wunsch. These are things that are talked about all the time, and most people sort of like nod their head and, oh yeah, I know what that is. But I think it's worth like actually seeing what the algorithm does, and appreciating the computational complexity of building this matrix and finding the optimal alignment. The last thing I want to say about this, one of the last things I want to say about this is that I hope this also gives you an appreciation as to why the hash-based mapping approach is so useful. Because if you have exact hash matches, those pieces you don't have to align, because you know they match exactly. So that saves a lot of computational time associated with figuring out the exact nucleotide by nucleotide registration. What you really just have to align using dynamic programming is the pieces that don't match exactly. Or you can at least use those exact K-mer matches from the hash that we use to essentially what's called seed an alignment. So it's like gives some basic structure to it, and then you're like cleaning up the ends with dynamic programming. And that's super helpful when you're aligning 150, or sorry, maybe a billion 150 nucleotide sequences. If we had to do hash mapping and then this dynamic programming algorithm for 150 billion nucleotides versus the reference genome, we would never be able to align DNA sequences for RNA-seq or for analyzing a cancer genome or a baby's genome compared to its parents in any reasonable amount of time, even with very fancy, sophisticated compute. Dynamic programming is crazy expensive. So a lot of people have expensive computationally. So these algorithms have been, people have worked on making this work on GPUs, graphics cards, and all sorts of stuff because it's complicated and time consuming. Okay, so as I said, this is a local alignment or a subset of the full sequence. Oh, the other thing, just quick note that I want to mention, this position here, we could get to 0 either by going this way or this way. And it basically, that's sort of a liner's choice as to what to do there. But in either case, because the alignment ends. All right, so as I said, global for Needle and Wunsch, local for Smith-Waterman. And so here's a concrete example of why this might differ, how this differs for larger sequences. So let's take this sequence, a shorter sequence aligned to this larger sequence. The local alignment is going to do something like this, where we just have this mismatch, and the rest are matches. These terminal subsequences in the larger sequence are omitted, but when we do a global alignment, we have to insert all these gaps and other mismatches to make the full thing match. All right. There's a lot of utility for a global alignment. This is often used for aligning whole chromosomes to one another, but for short subsequences, we tend to use Smith-Waterman alignment to find genetic variation and count reads for ChIP-seq peaks and stuff like that. Okay, so that's the end of today's lecture. What we're going to move into on Tuesday is we're going to talk about how DNA sequencing technologies work, and we're going to talk a little bit more about the FASTQ format. And I'm going to assign a homework on Tuesday that will be due the following Tuesday, just about mapping in the FASTQ format and some concepts, and get you working with those files a little bit so you're comfortable with them. So we have a couple minutes. If there's any questions, I'm happy to answer them. Okay, well, thanks everyone. I will see you Tuesday. And just a reminder that homework 3 is due tonight at 11:59.